Hi, everybody. I'm Terry Conneran with KRAS Kickers, and welcome to KRAS Cancer Connect about what's going on in KRAS. We're delighted today and to be able to bring in Dr. Kevan Shokat. And I'm not going to do a huge, long introduction about who you are and how amazing you are. Like we're all amazing fans and whatever. And if it wasn't for the drugs, like you're working on, but okay, but you're UCSF and like you're like the KRAS extraordinaire who found the way to do this. And oh, before I forgot, I have to make sure that I thank the sponsors that are allow us to be able to pay for this. And so thank you to Genentech Roche, to BMS, to Amgen, Veristem, and all the other amazing people that do this. But most importantly, thank you, Kavan, for doing this with us
Thank
today. And
you.
so thanks for being here. I'm going to just jump right in so that we can kind of get at it. I know we're recording. I'm not going to worry about people that do or don't make it in because I know you
Okay.
probably have to go through the disclosures and all that sort of stuff. But thanks
Yeah.
for being here.
Thanks, Terry. You hear me now?
I can.
Okay, great, great. Thank you, and thank you, Caitlin, for putting it together. And maybe just a little background. I reached out to Terry last week. Um, just always appreciate all the work you guys do at the KRAS Kickers. And I knew in the news last week there was a lot of issues of funding from the NIH, and really just wanted to reach out to you all to give you a little background on that, little update on KRAS, and I'll do that in about 30 slides, and then answer any questions you have. And mostly just wanted to be here to hear what you're thinking about and how I can help sort of us get through this stretch. So I will share. Okay, is that showing up?
Yep, it sure is.
Okay, super. Um, so yeah, we've been really interested in drugging KRAS for a long time, 25 years since I met Frank McCormick, and we've been doing everything we can to sort of engage that target. And I know that's such a central thing for the KRAS kickers. I've done a number of companies have come out of my lab. We discover molecules. We can't get them all the way to the clinic, so we patent them. The university licenses those to companies, and then we get those to the clinic. So that's kind of what these disclosures are about. Um, um, so we all appreciate that tumors arise from mutations in genes, very specific genes in our cells. And these mutations prop up in certain proto-oncogenes, genes that control normal growth. But when we get a mutation in one of these that locks them in a positive state, they give the cell an instruction to keep going through the cell cycle and to avoid the normal cell death that they might encounter. Luckily, that mutation alone is not enough to cause a cancer because we have protective mechanisms called tumor suppressors. These tumor suppressors sense that heightened growth. It's called oncogene-induced stress, and they arrest the cell by turning on a program of cell cycle arrest and pushing the cells to apoptosis. Now, those cells are now in a state of balance between the oncogene and the tumor suppressor, and that cell is also subject to mutations. And P53 is the second most, or is the most frequently mutated gene across all cancers. About 50% of cancers have a P53 mutation, and about 25% have the KRAS mutation. Once that synergy now has happened, the cell is going through the cell cycle very, very fast. And each of these red dots symbolizes an amino acid change and a mutation. And now you see on the right, after the cell is really moving fast through the cell cycle, it picks up other red dots. So it gets more and more mutations. Once the cell gets we don't know exactly, but and it varies on the site of the mutation. But 100, 1,000, 10,000 mutations, it starts to have the chance to alert the immune system because that cell is unique. It doesn't look like every other cell in the body. And so one of the protective mechanisms that the immune system has is it takes all proteins in a cell. It happens in all cells, chops them up, and if some peptides, parts of those proteins, have the mutation that was arisen in that evolution that I told you about, then it can be put onto what's called here an MHC class 1 molecule. So it's called major histocompatibility complex. It binds the peptide. And then that is what's presented to the immune cell, the T cell. And that green part of the T cell is very, very diverse, has like 10 to the 12th different forms it can adopt. And it tries to sense, is there a red dot on that peptide? Then I try to kill the cell. If it doesn't have a red dot, it looks like every other cell in the body. No T cell exists that can kill. So that's sort of the point of immunology. That's 40 years of immunology in 1 minute. So please feel free to ask me any questions about that. But that's the basics. But the tumor cell can fight back on the T cell. It puts up these proteins called checkpoint molecules. They have abbreviations, PDL1 on the tumor cell, PD1 on the T cell. That sends a negative signal to the T cell to turn off the killing. So even if all the recognition is there, the T cell gets an off signal. About 10 years ago, immune oncology came on more than that, 15 years ago, immune oncology came on the scene. These antibodies that inhibit the checkpoint inhibitors, they were developed, and that takes the brakes off the T cell, and now T cells can kill. So in this talk and in my lab, we're interested in therapies in all three categories. And I'll tell you about the KRAS targeting. I won't tell you about P53 work that we're doing, but I will tell you about immune evasion around KRAS. So just as a very brief background on why KRAS has been so hard to drug, it was the first human oncogene we discovered, but it wasn't until the two drugs I'll tell you about were approved in 2021 that we were able to get an inhibitor onto the molecule. And there are many drugs in signaling in these pathways. Most of them are kinases, and there are a lot of kinase inhibitors approved, like 70. And they take only about 9 years, 8 years to discover and develop once we identify the target. And the reason the kinases were so easy to drug is that they hold on to their ATP weakly. 20 micromolar is a weak number. 20 picomolar, a millionfold tighter, is a very tight number. So we couldn't use what we learned on kinases and apply that to KRAS. And people tried other strategies to prevent KRAS tumors from developing. And there was a very promising approach in the mid-80s called farnesyl transferase inhibitors. Those ultimately failed because although they blocked that modification, another bypass modification took over. So all of those trials failed. So we had a chance to treat KRAS-driven tumors back in the 80s, but because of biological backup mechanisms, unfortunately, those drugs didn't work.
Okay, can I
So,
ask a question? I hate to interrupt you.
no,
Okay,
I meant to say interrupt. Yeah.
okay. Oh, well, don't open that door. We'll be here 3 hours. But you should know that. Okay, but when you talk about kinase inhibitors, is that like what we're here about with the people that have EGFR for the TKIs, right? Okay, because
EKIs,
we
yes.
know typically, I know it's not always, but typically you're either EGFR or you're KRAS. It's
That's
typically
right.
you're not both. Okay. All right.
Exactly.
Okay, and so I want to make sure we're kind of like on following that. And so, okay, did I hear you correctly that those drugs, you can get them faster, you can find them faster to apply?
Yeah. Well,
Really?
you mean, I just mean they're easier to discover and
Okay,
they are like easier to go through all the stages of drug discovery. And then the regular clinical trial is no different. Is that what you
okay.
meant?
Oh, I just wanted, I wasn't quite sure how you meant that. And so basically you're saying I'm more complicated. Okay, so yeah, we could
Right.
upvote that. All right, okay, got it.
Yeah, yeah. Because that ATP in that middle of this slide is so
Okay,
easy to kick out, we have a drug that can fit there and we can make it, potent so that it fits in a pill, it's orally available, and
okay.
it's selective enough. Yeah, yeah.
Okay, all
But
right. And
because,
one more thing about the TP53 too. Okay, because you said
yeah,
about 50% of people that have cancer are TP53, and I know a lot of us have that as well. Okay, and you may or may not know how many people that have KRAS also have TP53. Is it more common or less common in us?
it's a great question, Terry. I don't know the number. I think it's probably, more than 50%. If it was just equal everywhere, it would be
Hmm.
50% of KRAS
Right.
tumors had a P53 mutation. I bet it's more like 70%, but that's just a guess. In
Okay.
colon, it's particularly another tumor suppressor called APC that is
Okay.
lost. So in each of the three, like we'll talk about the KRAS high frequency diseases, that's probably different. And I need to look that number up. It's a great question.
Okay. Sorry.
No problem. No problem. And so just to remind you how this KRAS works, it's really a protein that has two states, and one state is the off state, and one state is the on state. And it is off when it's bound to one nucleotide, a guanosine diphosphate, and then guanosine triphosphate is the on state. And the reason for the off is that these parts of the protein don't fold into the protein in the GDP state, but they fold into the protein in the GTP state. And then this surface is complementary to proteins that are in these signaling pathways, PI3 kinase and BRAF, and they get associated and turned on. So the whole cycle of RAS is really what the mutations are all altering. In order to go from the off to the on, we need to take out the nucleotide. And I mentioned it has really high affinity, so its half-life would be like months if it was not altered in any way. When you put this protein near it, a guanine exchange factor or a GEF, it reduces the affinity instantaneously and it falls out. Then GTP binds, and then basically it will be in this state and turn on what I told you about. Now, the timer is really the key to KRAS. In the wild type protein, it can be turned off by itself by this being water attacking the last phosphate and turning it to two phosphates. Or there can be this GTPase activating protein that comes up to the complex. And I'm going to zoom into that complex now. And you can see RAS over here and the GAP protein, GTPase activating protein. And now we're starting to get down to the molecular single amino acids. This is the glycine 12 in KRAS that is mutated most commonly. And when it's mutated to something other than glycine, glycine among the amino acids is the smallest. So any new amino acid is bigger and it pushes this arginine on the gap protein. So now the very fast hydrolysis is dropped out. So that's the basis for all of that transformation. The protein is locked in this state much more when we have that glycine 12 mutation. Now, very interestingly, across the different cancers, these are the 12 most lethal cancers in the United States. In green is the incidence, in yellow is the mortality, in blue is the frequency of patients that period of time would be KRAS positive. So in lung, it's about 30%, colon, it's about 50%, then breast, oral, prostate, very, very low, then pancreas, almost 90%. So that's the sort of distribution, and the glycine 12 goes to different residues in each of these diseases, in each tumor, and there's a distribution. So it's aspartate, valine, and cysteine, and these are three amino acids of the 20 that are in the human proteome. Then the
If we could
distributions.
pause right there, though, okay.
Sure.
All right, so when you're saying cysteine, I forget, it's not Valvoline, it's cysteine.
Veiling.
Sorry, too much television as a child. But that's where we get the abbreviations from. And you'll see those
Yes.
abbreviated as the CYS or the C or the V, or it's not actually A, that's actually
A,
the
it's
D.
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 Good point. That's right. These three-letter codes were kind of uh, the older ones, and now often when there's lots of amino acids in a stretch, you'll just see C, V, or aspartate is D.
Okay, all right, great. Okay, and so I just wanted to make sure we kind of like do that because it starts getting
Yeah,
convoluted.
right. Thank you. Yeah. So glycine 12 to cysteine, we often say G12C. So that would be this cysteine, or this cysteine. Yeah,
So it's not like one letter, it's just an abbreviation different.
that's right. It's just a
Okay,
different abbreviation.
okay,
Yep.
thank you.
Yep. Yeah, yeah. And so this is kind of the sum of all of the frequencies across all of those KRAS types. And of course, aspartate is the biggest, then valine, then cysteine, and then there's some more rare ones, more uncommon ones. And so when we started this project, we knew there was no pocket on KRAS that we could make it that we knew about. Nobody had ever identified one. So what we leveraged was a chemical reactivity of the new mutation, which is a cysteine. And we chose the cysteine of all of these we could have chosen because it is the most reactive. It's like the stickiest piece of super glue that we can take advantage of in all of the 20 amino acids in all of proteins. So we didn't pick the most common, but we picked the chemically sort of easiest one that we could start with. And so what I'll tell you now is a very short little intro to the drugs that target the cysteine, and then I'll bring you up to date with something we published just a little 6 months ago on the aspartate, which was a much harder chemical challenge and is more frequent. So when we started on this project, we solved the structure of KRAS with the cysteine, and you could see the nucleotide, which we've been talking about, and you could see these loops that change their conformation depending upon which nucleotide is there. In this case, it's the GDP right here. There's no obvious pocket on the protein, and that's kind of what we knew would be the case. So we went through a drug discovery approach that leveraged that yellow residue and that super glue. And we had, you could think of it as a piece of Velcro in our chemical library. And we kept trying out pieces to see what would fit around that cysteine. And that led us to this molecule and this crystal structure. And this crystal structure really broke, I would say, the whole field open because it identified this pocket. Because you can see the drug is sitting in a nice cave. And that cave nobody saw before because it was sort of where this flexible loop is. And either it was up or it was in this pocket. And so this piece of this drug helped us identify a sort of very, very fleeting shape of the protein that we had never seen before. And then it gave us a chance to make the ultimate drug and the drugs that are approved for this. Uh, and then it also helped us go after the other targets. And that's what I'm going to go tell you about. And I said also part of my reaching out to Terry last week was to try to tell you a little bit about what goes into this science so that you understand sort of what kind of equipment we use and sort of what we need when we say, we need a lot of sort of instruments and expertise to do this. And this crystal structure is the one, it broke open the problem. And we're very fortunate because right across the bay from us, so my labs are over here. This is San Francisco. This is the Bay Bridge. This is Berkeley. Cal Berkeley would be right here. This is Lawrence Berkeley Labs, and this is a cyclotron. This is where high energy physics happens, but also where we do X-ray diffraction. So this is a national facility, and people do different experiments inside this, and I'll take you inside. Inside there is this cavernous, very, sort of, you can't really go in here unless you know what you're doing. Otherwise, something will break. But in here is where we put our protein and we shoot x-rays and we see how they change their shape. And that's how we then back calculate what the structure is. So we really rely on, very, very big facilities all over the country that sort of maintain these facilities. And that's kind of what part of our sort of costs are and things like that.
It's not like you can just run this thing through an X-ray machine.
Right. Though the X-ray machines don't have enough power and focus. This one has many, many more, much more flux and much more pinpoint. And what we do is we take the crystal and we just keep pivoting it, and each time it shoots a crystal. So it's a much it's like probably on the order of 100,000 times smaller scale and 100,000 times more powerful. Yeah.
You're going to make me want to ask you if we can do a tour with you next year, right? That's
I think
coming.
we could, oh, in our lab or in this one.
Yeah.
Yeah, we could find out. I think
Yeah,
they do.
Frank asked me if we could do something, and I was like, well, if we can get Kevan in and we can do something at Berkeley, this would be like pretty cool. We're
I
doing
think it would
Canning's Lab
be.
this year.
Yeah, no, I think we could do it. It's pretty fun there. It's really
I would be, we'll talk later, but
okay.
like, okay,
Good.
but for sure, that'd be like pretty cool. Okay, so I'm sorry, go
Yeah,
ahead. But
no
you say
problem.
crystalline, let me throw this out there. When you say
Yeah.
crystalline, okay, again, the last time I had science and biology really, really was in high school. I had other things on my mind, okay? Really, I did. And when I hear that, I do think in terms of like crystalline, like rocks or or
Yes.
just water
Gems.
that looks like okay, like yeah. I mean, is that kind of like what you're
That's
looking
it.
at?
Exactly. Yeah, gems are, inorganic materials that crystallize in a very ordered shape. And then, how they have those beautiful trapezoid and sharp edges.
Mhm.
We grow a protein, which we do at UCSF in a facility, special facility that controls all the environment and stuff. Then we put it in a liquid nitrogen dewar, and they're frozen crystals, and they're held in here under liquid nitrogen. So when the X-rays hit them and they heat up, they're immediately cooled so they don't just crack. So it's under very important, very cool temperatures to keep it from
Wow,
breaking, melting.
cool. Very cool.
Yeah. So, we reported this molecule in 2013, and then we started a company, Araxes, that developed additional molecules. And this is what people call medicinal chemistry. They keep sort of adding and subtracting atoms and making it more and more potent. And as we get along, we see that this is, more potent. I'm not showing you all the data, but then this was the first molecule to show that we could shrink a tumor in an animal model. So very important milestone.
Okay, for us non-chemical people, can you explain to me what that thing is that I'm looking at? Okay, is this kind of like if I was doing like H2O, sort of one of those diagrams? Is this what that picture
Yeah.
is that you're showing to me?
Yes. Yeah. Yeah. So what's here is where the cysteine on the RAS will add. So the sulfur will push its electrons into here. These electrons will move to here, and then these electrons will go on to oxygen. So that's like the first thing that happens. Then those electrons come back here, they go back here, and then they come in and pick up a proton from H2O, from water, and then it's stuck onto the protein, which I'll show you another structure of.
Oh, cool.
So yeah. And then so you see that part is every one of these, but then we've been exploring the pocket and optimizing that binding to sort of get that to work better and better because it's a two-part thing. The molecule has to fit in the pocket, and then it puts this thing here right next to the cysteine, and then that's what slams shut. And so then other people and other companies began to also sort of explore this chemistry. They looked at these molecules, they designed a lot of things. So what I'm highlighting here is that all of them have that same place where the cysteine adds. And then they all have this ring, nitrogen, nitrogen in a six-membered ring. six-membered ring, six-membered ring, six. These are the kinds of things we look for to see that there are similarities, and that's the kind of thing. But then there's a little differences between them that I could tell you about. I'd have to show you more structures to kind of do it, but they're all fitting in the same pocket that we identified, and so that's very satisfying to have that discovery just keep playing out. And these are the two approved drugs right now that you all probably heard of.
Kind of a
Sodarac.
Tetris sort of a game almost.
Exactly, exactly.
I'm sorry, I have to just dumb it down.
No, that's perfect. I'm glad yeah, that's great. Um, so one thing we've been talking about, you asked about the structures, and I tried to point out like, okay, this is different than this. One thing very important for us is when we make a molecule, it's like cooking. We put it in a flask, we add some heat, we add some reagent, but the final molecule, we need to know it is what we draw here. And to
Mhm.
do that, another instrument that we use is a nuclear magnetic resonance spectrometer. And this one is in the bottom of our building, which you visited, Terry. And we take the molecule in a solution, and it's in a very small probe. This is about 10 millimeters across. And you see this solvent there at the bottom. You can see the meniscus, the top of it. And the molecule is very concentrated. And we put it and lower it down into this magnet. And then we pulse it. And then we also measure how the resonances of protons move. And that we see a spectrum like this, and then we can assign it, and then we know we have what we think we have. And this is about one of about 7 instruments we have in our building. And these are so essential because we use it every day to confirm our structure. And these kinds of, oh, sorry, one thing. 12 o'clock checkout. 12 o'clock checkout. Okay, all right. Thank you. No matter how many times. Okay.
No worries, no worries.
Um, so this is like, we have to keep all of this is a big magnet. We keep it cooled by liquid helium. So, this is kind of just telling you there's a lot of things that are maintained for us to do our work. And that's kind of what I'll get to at the very end about how we cover grants and things like that.
Okay.
So I've updated you on the cysteine, and this is one of the clinical molecules. And a lot of people are kind of moving to other residues, and they're taking off the piece that reacts with cysteine, and they're making ones that inhibit all of the variants. But I have been trying to keep making molecules that are specific for each individual amino acid. And so to do that, we had to go to harder and harder chemistry, which because these are not as reactive, they're not as tight of a glue,
Mhm.
super glue. So we made some for serine and arginine and aspartate. And of all these, the aspartate was not only the most common, but it's the hardest because its reaction is the weakest. So we worked on this for about 15 years. And last year we were able to publish our molecule. And
Okay, one more time, wait, go back one slide. Okay, so the aspartide is D, okay, the
that's
cysteine
right,
is
D.
C, okay, the
Yep.
V is victory, okay, now I've got Valvoline in my head again, I'm sorry.
Valine,
Valine, okay, the SER is that,
serine,
serine is that S,
G12S, yes.
okay, and is it
Ar
arginine is R?
Arginine is R, good.
Okay, all right, and the other one is A? I don't know what the
It
actual
is
name
A.
is
That's
A.
right.
Okay.
Alanine. Yep.
Yeah, I actually at one point had a cheat sheet on my desk, and
Yes.
mostly I can nail them now. Okay,
Oh, very
and
good.
oh, well, I can be sharpened up. Okay, so it's from hanging out with you. Okay, so but looking at those things, each one of those has a different level of stickiness and different level of problem. And is that one reason why we have certain ones of those and G12D is the ASP. Somebody just posed that question. Is that one reason why the different level of stickiness is why there's more or less of cancer in those places?
No, we don't think so. And the best evidence for that is the cysteine kind of residue is kind of it's caused by a different path of mutation. So it kind of has to do with what is sort of generating the mutation itself. And we we have studied a lot of why certain residues come in certain cancers. And one of the most outstanding questions that had been around for a long time is why arginine, or R, G12R, is really only in pancreatic cancer. And Channing Durr, who you're going to go visit his lab, he was the first one to pose a really viable answer to that question. So we keep wondering a deeper part, the answer to your question, and people keep looking for it. So it's not about the chemical reactivity quite. Yeah.
Okay, okay, but okay, so and one more time, the cysteine CYS is CG12C, the
Yes.
ASP aspartide is D. We have
D.
no idea why they called it the, I guess the A was taken. I'll just make up my own story to keep it interesting. Okay, then we've got V, S, R, and A, right?
Yes. Mhm.
Okay, perfect. Okay,
Yeah.
thank you.
And so, yeah, so we discovered this, which is a novel chemical group that allows the aspartate to add covalently. So like we did on the cysteine. And once we discovered that, we were really amazed because it works. Now I'm going to the single letter things. So it goes to G12D and labels at 100%. G13D, the next position, is also sometimes mutated, but it doesn't work. This drug won't work on the 13. It's so selective for the 12. And even though wild type RAS has 26 other aspartates and similar reactions, amino acids, glutamate doesn't react. So this was a real milestone for our lab. This was the thing we waited 12 years for. We worked on for 12 years to get. And we wanted to again understand, how does it fit in the pocket? And we solved its structure. And the key part was, now we see the bond between the aspartate and the drug. This is the first time anybody ever saw a bond between this weak amino acid and the drug. So it really proved that we could actually grab onto it and hold. And
That's
so,
powerful though, okay, but I need to really recognize, okay, and honestly, okay, I'm not just a G12D patient, okay, but it's the bulk of us out here, okay? And so the fact that you stuck with it, you kept looking at it and you found it, and okay, we don't have a perfect solution yet, okay, but you can't do anything till you find it. That's
yeah,
that's amazing that you actually have been able to target and pinpoint into it. So thank you for your work and all the people that you guys are doing, but I'm really excited to
yeah,
see where it's going and what I
yeah.
see what's
No,
happened with C so quickly.
yeah, I mean, even though we worked on it for 10, 12, 13 years, I honestly thought it would take until like 2035 to get to this point. I was willing to put in 25 years to get to this point. So in my book, we're even ahead. So it's really, that's why I'm so excited. Yeah.
I'm excited too, but you see, I sound excited, I get enthused, I get like very dramatic, and you kind of like, oh, I'm excited. You're much more cool than I am.
And so this, when we're trying to sort of understand how drugs work, now we're using, you hear it a lot in the news about artificial intelligence and, AlphaFold predicting. So a lot of our things are going more and more in these computer directions. And so that's something we have to redesign our rooms to have more cooling and things like that. So as we keep up and new advances come, it costs us more money in the university to sort of use those things. One of them is computers. This is something might be too detailed for some of you, but I really wanted to bring it up because as we're making molecules, this is one we made, this is another one we made, this is another one we made, and we highlighted the chemical differences. So these are the kinds of stepwise things we do. And when we put these into cells, we know they each bind to the G12D, but they might do it a little better or a little worse. And one thing we use is a measure of all the sort of changes inside the cell the drug induces. And I think one of the best signals, I should have just cleaned out a lot of it. I want you to focus just on this. This is the measure of the G12D protein.
Mhm.
And you can see that when we add this drug and we treat it for up to 12 hours, we reduce it a little bit, but not that much. The next molecule is a little better. We reduce it more, but not completely. This molecule shocked us because you see in the first hour, 2 hours, it's gone. The, the protein has been completely changed because the drug is on all of it. And one thing that we kind of wondered, and this was the postdoc Chen Heng's idea, was he said maybe this molecule binds to the GTP state of the RAS. And
Mhm.
remember, the GTP state is where the mutation locks it into, but we know it cycles sometimes. So these molecules are weaker to GTP, and this one is stronger. That was the prediction. And here in red is the GTP form, and in black is the GDP form. So these two drugs, they modify. This is time. This is modification. This is a maximum 100% modification. If we put the protein in the GDP state, this one labels it. GTP state, it's not so good. Oh, this one's better on GTP, but still on GDP. But this one, remember the special one, it flipped. It prefers the GTP state.
Wow.
That was a real surprise to us. And I was speaking at a medicinal chemistry meeting 2 weeks ago. And I put this in because I feel like it's a very surprising thing. It opens up a window and an avenue to us to make even better and better drugs. And I wanted everybody to appreciate it because it was buried in a paper of ours and maybe didn't catch people's attention. So, but it's just to give you an idea of how this works and how the surprises come. And sometimes there's good surprises. Yeah.
No, that's an awesome surprise. And the fact that it's actually not just like a little responsive. I mean, it's like, man, it's
Yeah,
rocking it, right?
exactly.
I mean, and so
Exactly.
that's what leads to not just theories, but to actual drug development.
Right. And that's the kind of thing, to be honest, right now with computation, we can't do always. That's why we need to make molecules and test them because there's no way a computer could have predicted this. This is just too subtle, you know? Yeah.
That's pretty cool. That's pretty cool, though. I've already found that just from my little bit of playing with AI, it's like, oh, wow, how quickly I became a slave to it. You still actually need to do it in the lab to see what's going on. And
Yep.
as I've heard once, if I haven't heard a million times, we've cured cancer in mice, but we, often, of course, we've also caused the cancer in mice. And so the problem is we need to kind of get it from there to the mice to the people to actually get it upscale it, I guess.
Yep. Exactly. So that's kind of our work. I wanted to tell you about and tell you, I told you about the cysteine and the approved drugs, and I told you about our covalent drugs. And there's a lot of exciting developments. I know you guys cover a lot, and there's a lot of new ones coming. One I really wanted to highlight, which I know you've heard of, is the Revolution Medicines one that I'm a co-founder of, but I didn't explicitly work on this project with them, that they've got a molecule that has got this name now. The actual one in the clinic is 6236, and it binds up in a slightly different area of RAS, and it uses this other protein to actually bind to RAS. So this is a drug that binds to one protein in the cell and then binds to this protein. And when you sort of put all the surface around, you see that the drug is buried inside there. And if we zoom in now you can see RAS here recognized by the RAS drug, the RevMed drug bound to the cyclophilin molecule. And basically, it's now we're recognizing RAS using another protein in the cell that we've redirected with this molecule. So it's I think the one that's in phase 3 for pancreatic cancer with a number of the mutations, all of the mutations. So I wanted to give you one flavor of that, but there's a lot more coming, and cover those. Yeah.
Yeah, no, it's a really exciting, some of the stuff that they've got going and moving into
Yeah.
the phase 3 trials as well. We have a lot of people that are benefiting from it. And I love the fact that, and I'm going to mention this just because it's on screen. There's an opportunity. Okay, whenever something ends with an IB, it means it's an inhibitor. And people have, and the companies are really trying to get like the RAS or something in the middle of it. And so we've got the RAS SIB, so it's a RAS inhibitor. Okay, and this one is like, this one's on, right? And so it's a RAS on, okay? And so if you work it backwards,
Yes,
it's
exactly.
not the random alphabet that we think these drugs are, okay? And the same thing with adagrasib and
That's right.
sotorasib because it's got the
That's right.
RAS and this IB in it. And so I'm just bringing it up because
No,
it's like
those are good.
until you find this stuff out and you're like, oh, I mean, otherwise we just think you guys are sitting around, you just throw letters into a box and just pull
Hold
them
on
out.
1 second,
Sure,
Terry.
sure, sure. And you guys, they don't actually do that. So anyway, a little off topic, but of interest. I appreciate with him, he's in a hotel room in Santa Fe right now, and so he's had a brief interruption. Okay, no worries, no worries, we're all good. So, okay, no, that's really exciting, and the fact that these things are happening and so rapidly, we're actually benefiting as patients. And like you said, I mean, this is what, a solid 10 years before you thought it would even get to this point. That's amazing.
Yeah, yeah. No, I think well, I think Terry, I should skip this end part because I want to make sure I have time for questions.
Okay, go for it.
So I'm going to skip that part. I can
You
come back to
sure?
the time. Yeah, yeah. I think it's just a little too much. I wanted to end with this and then make sure we have
Okay.
15 minutes for questions. Does that sound good?
It sounds good. If we don't have any questions, we'll go back to the slides.
That's good. Okay. So I wanted to cover this that, we've been hearing from NIH and the federal government that this indirect cost recovery is too high on our grants at universities. And I wanted to give you a little background on what goes into these costs and how we budget things. So direct costs include like the materials that we use on this project, the reagents, the chemicals, the glassware we have to buy, salaries for the people, little instruments, computers, cell culture, travel to meetings, tuition for the students. Indirect costs are things that are like we need for this project, but we use them for other projects as well. So we sort of divide up and spread that cost over multiple grants. That's laboratory space, electricity, air handling for chemical hoods. So we can work with toxic chemicals, tissue culture, NMR I showed you, environmental health and safety. So we get inspected. So everything's working well. And and then like just the administration of getting the money in, spending it correctly, telling them we spent it right. And I made this little dummy budget to give you an idea because the two numbers we've been hearing about are, a bit, I don't think that we people quite see what's behind them. And we've been looking at it at the university too, because we want to get any waste out and everything. So on a typical NIH grant, we have a indirect cost recovery. It differs by university, but I made it a simple number, 50%. At UCSF, it's 58%. And if we get direct costs like salaries, reagents, small equipment of $1 million, NIH gives us $400,000 in direct cost to cover those other items. So the total is like 1.4 million. On a private grant like the Gates Foundation or something like that, they've negotiated a 15% rate. But there's a little bit of a catch, which is you'll notice that this 50% should be 500,000, but it's 400,000. That's because indirects don't go for the whole amount of directs in NIH. Equipment doesn't count for that. So it's always less than that amount. So the total is 1.4. On the private, whatever the 15% is, it's applied to the whole grant. So it's really 15%, 150. But the other wrinkle is that within this direct funding in a private, a lot of the indirect costs are packed into this number. So the actual amount for salaries, reagents, et cetera, is reduced. And it really is actually sort of money in indirects. So overall, even though these 2% are different, the way directs are calculated are kind of different. So just to give you a little bit of an insight into that, we can go to questions now.
And I have an accounting background. And okay, yeah, and one of the big surprises to me in labs in general is they kind of expect y'all to have some sort of business, like real like business accounting, sort of like management to be able to come up with budgets and direct indirect costs and all this sort of thing. And so it's always been kind of like a bit of surprise. And so I think that's very helpful kind of going through, it's I did a lot with like property management. And so there was like common area costs. And for a common area cost, okay,
Exactly.
it applies to everybody, everywhere, okay, whether it's like, in the building, okay, the elevator is there for everybody to use. And so it's a common area cost. It's not just a cost to the people that are on the second, fourth, fifth, and 12th floor, whatever, okay? And so it's actually a common area cost. And so that's kind of like the indirect cost compared to the other stuff. So anyway, yeah. Um, okay, I'm going to see if there's any questions from the audience. What we've got going on. Okay, all right. So do we have any questions from the audience? I can see that Annie is here, and Annie loves asking questions. Let's see, we have one. Hi, have you tried KRAS targeted therapies on any patients prior to chemo? I think we may need to get some clarification about where your role is. And so let me read the question. I'm
Oh,
just kind of reading this, like reading this live, okay? And then we'll kind
yeah.
of back into it, okay? It
Yeah.
says, hi, have you tried KRAS targeted therapies on any patients prior to chemo? Was the response better or the same, similar to neoadjuvant immunotherapy? And some background on her dad. Okay, if you kind of want to address the difference between what you see versus like an oncologist.
Yeah. So I'm a, PhD chemist. So I don't treat any patients or really sort of manage any of the clinical trials. So that's all done by the company's chief medical officer and the teams in participation with the investigators at the universities that run the phase 1 and phase 2 trials. Yeah.
Have you worked within your lab any, like say, tumor samples from actual patients that you guys have turned into slides and stuff?
We've done that a little bit, especially with resistance models, where maybe somebody at UCSF was on one of the drugs, then became resistant, and then we use the samples to try to test ideas that we have for how to overcome the resistance. Exactly.
Okay, all right. And then again, I just read these as we're going through. It says, how
Sure.
much time or effort does it take to be compliant with the federal grants?
Yeah, it's a great question. It's really increasing more and more over the last 10 years. Um, and there's just been more check-in points, more watching line items that we're not spending, money on the wrong expenses and that they're all lined up. A person on this grant is only paid on that grant. And yeah, so and we used to be able to, for example, buy one piece of equipment on one of our grants. But if that piece of equipment is going to use for other grants, now we have to parse it apart. That was a very new thing just 2 years ago. So we have a lot of compliance. And when the NIH reviews us, that puts at risk the entire University of California, San Francisco.
Now, are you actually doing that, or do you have people that can help you out with that
Oh
sort of
yeah,
thing?
I have a what's called a post-award person, Angela, and she's got fantastic, she used to work at Lockheed Martin and then came here about 10 years ago. So she's got government contract, really good. And yeah, it's one thing I try to do with all the people like Angela that really work with us is after we go through my budget and we spend about an hour line by line, whatever, I ask her, Angela, what do you want to hear about from our lab? And I give her a menu of Parkinson's disease, cancer, inflammation. And she says, I want to hear about this. And so I go to the whiteboard and I tell her about the actual science because she's so valuable to us. And I want her to know what's behind the numbers. And it's very, very fun. I started that a couple of years ago.
It makes a big difference when we have a sense of understanding around it, as you know, I mean, it's not like I'm going home and I'm making this stuff. I mean, I have no idea what you're doing. Okay, again, I'm going up next down the questions. I see two questions around G12R. And so we have members who want to know why KRAS G12R has less options.
Super, super question. It is because that residue, that arginine, it really is very charged and it folds back on RAS and it holds on to GTP tighter. So it's one of the least cycling of the mutants. And so a lot of our drugs bind to the GTP state, like I told you that little story. And R is like the most extreme of the GTP state. So a lot of the drugs that are evolved from the G12C drugs, they don't work very well in G12R. Um, so that's why the RevMed approach is probably the first one to succeed there. But at this meeting I'm at, I just heard from a group at Boehringer Ingelheim that they've got drugs that now work on R. So that paper should come out soon. So it was harder, but it was it wasn't, there was no fundamental reason we can't do it. It just took a little more time.
Yeah, and they just had a conference last week on Boehringer Ingelheim. They had a conference I think it was last week, on targeting the pathway, and I wasn't able to attend the whole thing. I got part of it. Very cool. All right, and then let's see. How are testing with arginine for G12? How far along is the testing with arginine for G12R outside of RMC 6236?
It's preclinical, but I would say usually when we're at this stage, from what I've heard, then it can make it to the clinic in sort of 2-ish to 2.5 years, phase one. That's a very rough,
And 2.5 years sounds good when you're dealing with
No,
anything
I
having to
know.
the government and with life
Yeah,
expectancies being as sadly low as they
I
are.
know. That's with
And
just that
yeah,
approach, though. Like I said, the RevMed
yeah,
approach works right now. So
yeah.
that,
That's
yeah,
really exciting what they've got and what
that's right.
they have happening.
Yeah.
Let's see. Actually, right up that. On RMC 6236 and 9805, are these binding different sites?
No, they bind the same way, but the molecule has a little, in 9805, it has a little handle to make a covalent bond to the aspartate. And it also weakens its binding to other mutants, other form, the wild type. So it's more specific and selective. Yeah,
Okay, when you say wild type, you mean not mutated.
that's right. Not
Okay,
mutated.
right, okay. And again, I'm just doing these in order. Do you have any sense that the new trials will be affected by the changes in the NIH policy?
I wish I knew more. I think there's just a lot of changes like reducing the staff at the FDA. That'll slow down, inevitably reviewing things, and that slows down all the steps of, safety testing and things. So I think unfortunately it will, but we don't know the degree. Everybody is so motivated for these drugs that it's going to be, working harder and harder to make sure we don't lose any time, but we don't know. Yeah.
Yeah, and and part of it is that like a lot of the funding does come through like NIH and then it's in all the the government facilities, but a lot of it is coming through sponsors as well, through pharmaceutical companies. And I think that's one of the things I was so completely unaware of, like coming into this, that it's not like it's not all one pool of money that's
Right.
coming out there. And so when we see stuff in the news, it's not necessarily taking everything into consideration, it's taking one thing into
Yeah.
consideration.
Yeah.
And it's made me a lot more, I've gotten much better about like looking past the headline, I guess.
That's
Um,
kind of what I was hoping. Yeah. And like getting the drug to the approval and getting it to patients, like I was trying to show, there's so many steps. And yeah, like you said, no one funder pays all the steps. But if somebody who's been participating drops out, it's gonna really That's the worry.
Well,
That's the
and
worry.
we're Americans, okay? And I know you don't work for $0 any more than anybody else does, right? And so at the end of the day, if there's money in it, there's more motivation to do something about it. And I
Exactly.
know that that's kind of an ugly way of putting it, but it's nonetheless, it's the truth.
Yeah.
We're not going to solve the fact that we are being overrun by dinosaurs right now because there's no money in it. We don't have that problem.
Yeah,
So,
that's right. That's
and
right.
so I don't know. I voted, and so I try and stay out of policy.
Yeah, no, I'm not. I'm not here to tell you about that. I just, yeah, I wanted to
Yeah,
give
yeah,
you.
yeah. I know. I know I'm not fighting dinosaurs today. I try to comprehend the on-off states. Do you recommend any literature to learn more?
Ooh, um,
Hmm.
yes, there
I'm
are
actually
great
thinking about
papers.
doing a class. I'm actually thinking about doing like a class and doing that
Oh
in
yeah.
that stance.
Oh,
And
good.
of course, you would be on my list of people to bring in.
Yeah, yeah, I'm happy to. And I think in a lecture where you could ask questions, it's better than the reading. The reading, it's it's dense and a lot of it, it's hard to pull all the threads together. So yeah. But like I said, I think any of the pieces uh, by Channing, Frank McCormick, they've written very good pieces on RAS that cover that. Yeah.
And just shout out to the KRAS Kickers. We have a KRAS Kickers YouTube channel, and there's a lot of information, videos on there that are going through your previous presentations with us. We've got Channing's previous ones. I can't even begin to tell you, Bob Lee and Pasiani. We've got a number of them on there, and I gotta say, I mean, I've been privileged to just be surrounded by such generous people with your time and your knowledge and sharing it with us because we just want a sense of understanding for something that we're
Yeah.
just trying to fight against, right?
Really,
So I
really
really appreciate it.
appreciate all of
And
your
and I'm
guys' questions, and they're so timely and they're so prescient, I think. So yeah,
we're kind of
I better
desperate.
wrap up in a minute because
I know we're going to wrap up. I'm scrolling through these really quick, so I'm going to be really fast, okay?
okay.
Is there also anything for KRAS 13D or
Yes,
now or soon?
there is RevMed drug coming down the line for 13D specific, and then also 6236 works in there too.
Okay, I saw at AACR, RevMed on off had response to BREF V600E, post use of Beacon with an EGFR inhibitor. Do you see future with RAS on off with KRAS Wild patients?
Too complicated for me to answer that one. I'd have to. That was a lot of
Yeah,
mutants.
I kind of thought that was a
Yeah,
lot for a last minute thing.
yeah,
Okay,
yeah.
okay, G12R, another G12R question. A lot of thank yous. Do you have any potential for revision to control? Oh my gosh, Annie, I love you. She's asking very complicated questions. Do you see any potential for a revision to control arms for phase 2, phase 3 trials? Patients aren't usually interested in control arms. This is a massive problem.
That's a really
And
great question.
so less spots are open to US patients.
Yeah,
Dr. Azad suggested a pooled control arm to get historical data for KRAS trials. I personally have talked to the FDA about this, and
yeah,
it's aggravating.
yeah. I would really get the clinicians. I'm really not the right person. Yeah,
Yeah, so it's frustrating. And thank you so much
yes,
for reaching out last week. And I'm going to hold you to that whole like let's do something like next year. And
sure.
Frank threw it out there. I threw out the gauntlet. So I'm
Oh
going to pick it
yeah,
up and I want to like I want a whole
good.
party in San Francisco. Let's like make it happen next
Perfect.
year.
Let's
All right.
do it.
Thank you so much for doing
Thanks,
this,
Karen.
Kevan. And you have safe travels home. We hope to see
Thank
you
you.
again real soon, like AACR or something.
Yes, I'll see you there. Okay.
Okay, thank you all for joining us today, and we'll be ending here and putting this on a video soon. Bye
Take care.
now.
Be well, everybody.